Blood vessels and endothelium. This video is part 1 of the series on blood vessels and endothelium in Prescribon Medical Education series. See also Prescribon Medical Education series on atherosclerosis, part 1-2. Blood vessels range in size from microscopic capillaries to large vessels such as the aorta and the vena cava, and vary in specialized function from tissue to tissue. They deliver oxygen and nutrients, remove waste, control the passage of cells and macromolecules from the blood into the tissues, and are equipped to sense and respond to physical and chemical signals. There are three basic layers to blood vessels: the intima, the media, and the adventitia. Arteries are divided into three types based on their size and structural features. Large or elastic arteries, including the aorta, the major branches of the aorta, and pulmonary arteries. 2. Medium-sized or muscular arteries comprising smaller branches of the aorta, example coronary and renal arteries, and 3. Small arteries less than 2 mm in diameter and arterioles between 20 to 100 micrometer in diameter. The 1 micrometer is 1,000th of a millimeter, and arterioles of this size within tissues and organs. In this picture, we show a structure of the artery wall having three main layers. The inner layer adjacent to the blood circulating is the tunica intima layer shown at the bottom, followed by on the outside by tunica media layer, and the outermost layer, which is in connection with the surrounding tissues, is the layer called tunica adventitia. We have seen the structure of artery wall in the previous clip. Now we will see all the three layers of the vessel wall in little more detail. The intima, that is tunica intima, comprises a single layer of endothelial cells on a basement membrane, beneath which, depending on the vessel size, there may be a layer of fibroelastic connective tissue and an internal elastic lamina that provides both structure and flexibility. Embedded in the intima are pericytes, intriguing cells of smooth muscle cell lineage that make contact with multiple endothelial cells. The media, tunica media, is made up of predominantly smooth muscle cells and concentric elastic fibers making up the elastic laminae. The outermost part of the vessel is the adventitia, tunica adventitia, a less well-defined layer composed mainly of fibroelastic tissue that provides structural integrity to the vessel, but also contains nerves, small feeding blood vessels, they are called the vasa vasorum, and lymph vessels. However, the adventitia is also in continuity with perivascular adipose tissue, PVAT, that has paracrine relationships, that is, allowing cells to communicate with each other by releasing signaling molecules that bind to and activate surrounding cells with the vascular wall. In simpler terms, the intima may be considered as the layer that transmits signals from the lumen of the vessel, that is from the blood, to the rest of the vessel wall and controls the interface with the blood. The media is the mechanical workhorse of the vessel, and the adventitia links the vessel wall to the local and wider environment. Dear listeners, by the way, This educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Now, coming back to our discussion regarding blood vessels, not all vessels have each of these three layers, and the layers vary in size and structure between vessels. For example, capillaries are essentially endothelial cell tubes surrounded by pericytes. Resistance vessels have a relatively thick media, and the large conduit arteries have a high proportion of elastic tissue and a rich vasa vasorum. In disease states, particularly atherosclerosis, the vessel wall may have a high content of inflammatory cells in the intima, media, and adventitia. All three layers of coordinate to regulate the function of the blood vessel, and all three are involved in the pathogenesis of vascular disease. Large conduit arteries perform the function of mass transport. Smaller arteries and arterioles provide the predominant resistance to flow and are therefore key determinants of blood pressure. This figure shows the structure of a capillary. Capillaries are thin-walled and contribute most to passage of nutrients, gases, and cells through to the tissues. Venules provide post-capillary resistance and help determine capillary pressure. And larger venules and veins dynamically regulate the total capacitance of the circulatory system. Endothelium. A monolayer of endothelial cells line the intimal surface of the entire vascular tree, arteries, veins, and lymphatics to form the largest endocrine or paracrine organ in the body. The endothelium is a spatially distributed organ, that is, it is situated in so very many places. The endothelium weighs approximately 1 kilogram in the average patient and covers a total surface area of 4,000 to 7,000 square meters. Recall the figure of structure of artery. Endothelial cells are metabolically very active and exert a profound influence on vascular reactivity, that is, response to various stimuli. They have a role in thrombogenesis, that is, clot formation. They have a role in coagulation, that is, solidifying blood, and the behavior of circulating blood cells. Abnormalities of endothelial function have been implicated in a wide variety of diseases, ranging from atheroma and hypertension to acute inflammation and septic shock. Note: thrombogenesis is the initiation of a blood clot that forms on the wall of a blood vessel or in the inner heart when blood platelets, blood proteins, and cells stick together. A thrombus may block the flow of blood in a vessel. Coagulation. Coagulation is the process of blood clotting, that is, the process of platelets, that is a type of blood cell, and proteins in the plasma, the liquid part of blood, working together to form a clot over the injury. Anatomy of the endothelium. Each endothelial cell is between 25 and 50 micrometer, that is 1 micrometer is 1,000th of a millimeter. Endothelial cell is 25 to 50 micrometer long, 10 to 15 micrometer wide, and up to 5 micrometer deep. and lies with its long axis aligned in the direction of blood flow. Recall the structure of artery wall earlier. The underlying smooth muscle cells lie radially or about 5 to 10 micrometer wide and taper at either end so that a single endothelial cell can communicate with many smooth muscle cells and vice versa. The endothelium also comes into intimate contact with circulating cells, and the total area of the luminal surface of the endothelium is in excess of 5000 square meters. This thin layer of cells is particularly susceptible to injury and changes in endothelial cell morphology and turnover as occurring in hypertension, diabetes, and atherosclerosis. Signal detection by endothelial cells. The endothelial cell membrane has many receptors for circulating hormones, local chemical products, and vasoactive substances released from blood cells. Endothelial cells sense pressure, stretch, and blood flow via a number of different sensors that vary in different vessels, reflecting the differences in pressure and blood flow across different vascular beds. The endothelial cells in vascular damage and repair. Vascular endothelial cells move in response to specific chemical signals and can migrate to recover areas of endothelial damage or denudation, that is loss of cells due to various factors. Circulating endothelial progenitor, that is precursor cells, EPCs, derived from bone marrow are involved in the processes of vascular repair and the response to tissue injury. These cells are characterized by specific cell surface markers CD34 and CD133 and can form colonies of many cells. There is also evidence that resident stem cells located in the vessel wall with properties of clonality, self-renewal, and multipotentiality, that is, ability to form different kinds of cells, can replace local, damaged, or denuded, fallen off endothelial cells. Note, stem cells, these are cells from which all other cells with specialized functions are generated under appropriate conditions. Clonality, that is, a group of cells or organisms that are descended from and genetically identical to a single parent, such as a bacterial colony, whose members arose from a single original cell. Pericytes. Pericytes are long cells, approximately 70 micrometer, with extending cytoplasmic processes around endothelial cells that make multiple cellular contacts. See figure structure of capillary earlier. The junctions between pericytes and endothelial cells appear to be rich in growth factors, particularly epidermal growth factor, that are important in regulating endothelial cell growth and may be vital for angiogenesis and inflammation. Pericytes can also differentiate into other cells such as fibroblasts. The nature of the junction between pericytes and endothelial cells may be important for regulating permeability at special sites such as the blood-brain barrier and in the response to ischemic injury. In other areas, the contractile function of pericytes may predominate. In the retina, where pericytes are particularly prevalent, their loss is associated with diabetic retinopathy. In inflammation, neutrophil transmigration from venules is regulated by adhesion to pericytes. The functions of pericytes are listed below. 1. Contractility. 2. Barrier function and regulation of permeability of various blood substances. Neutrophil transmigration in inflammation. Control endothelial growth and new vessel formation, sensing hypoxia, that is decreased oxygen, and hypoglycemia, that is decreased glucose levels. They can transform into fibroblasts, which produce collagen and elastin fibers in wound healing, cancer metastasis, and they can differentiate into smooth muscle cells. Dear listeners, here comes the end of part 1 of this series. For part 2, see Prescribon Medical Education series, Blood Vessels and Endothelium, part 2.